Thank you. So welcome back, everyone. Another light here and another one. Okay. All right, always in the course on bioethics in the Masters on Molecular Biology at St. Thomas University. We're going to be looking at today stem cell research and cloning, human cloning. Beginning always with a little prayer in the name of the Father, the Son, and the Holy Spirit. Come, Holy Spirit, fill the hearts of the faithful and kindle in us the fire of your divine love. Amen. Okay, so you've noticed we've made the transition now into healthcare, bioethics, okay? And it covers, it hovers around today's topics, even though it's within healthcare, it's also beginning of life issues, because we'll go back to especially the sources of stem cells that make it either ethical or not. So we'll look at stem cell research, distinguishing between human and non-human research, then distinguishing between adult and embryonic research, and some alternative sources of stem cells, cord blood, pluri-iPS cells from Yamanaka. And then we'll look at cloning, the two types of cloning, reproductive and therapeutic cloning. Without further ado, go forward. There's a theologian and philosopher from the Middle Ages, Thomas Aquinas, not Thomas of Villanova, who is the patron of this university, the Augustinians. Thomas Aquinas, he was a Benedictine monk. He was a great Catholic theologian and wrote a lot about moral theology and ethics. There's a saying attributed to him. He would say, never affirm, seldom deny, and always distinguish. So in ethics, we do a lot of making distinctions, right? So sometimes if I were to ask the question, what is your sense that the Catholic Church is in favor or against stem cell research? I just frame it like that, stem cell research. The church in favor or against stem cell research? Either one, either yes or no. Anyone else? What do you think? No? Stem cell research.
I'm sure some forms are cool and some yes.
Right, that's the thing. What kind of stem cell research do you mean, Professor? Because there are different kinds. And so it's all about the various types of research, right? So we have to make these distinctions. And with regards to stem cells, we have to make at least two levels of distinction between human and non-human, and then within human, between embryonic or adult stem cells. And that's the key, at least for the bioethical aspect of it, okay? So again, the first level of distinction is, are we talking about human stem cells or non-human stem cells? Because practically every species has stem cells, every animal, right? And especially the mammals, because it's part of the regeneration tissue. If I cut myself, then cells will come from the inside, mostly the adipose layer and the dermis, to replace the damaged cells, all right? And there's a healing that occurs from within. And who is producing or what's producing that new tissue? Stem cells are producing the tissue. So we all have stem cells, and other animals do too. And there has been a lot of research done with mouse. Of course, the lab mouse, Mus musculus, is the one who usually gets it, including embryonic. But those would be non-human stem cells. And with all due respect to animals, different ethics or applies, less rigorous in the sense that what we may do to animals, we may not do to humans in certain realms, especially if it harms them significantly, right? While I'm on this tangent, how do we justify harming animals in the lab? For the purpose of what? Typically, medical research, okay? In other words, the ultimate goal is, for example, to test a new drug to see if it works. Before we go to clinical trials on humans, there will be animal trials, all right? So we'll test it on the mouse or the rabbit before we give it to a human. And if it's showing still toxicity and whatnot, that will be worked out before going to the human trials. But even before we test on mammals in the lab, we will test on invertebrates, maybe the fly, the fruit fly, okay, which can be raised in thousands. And perhaps even before testing on the fly, we could test on the bacteria, which now we're at the level of millions, okay? So there's always that protocol that we begin with the inferior animal first, or the inferior species first, okay, inferior by way of maybe less behavioral, and yeah, especially if there's not a nervous system, right? Or if there's a nervous system, a more primitive one, just in case they are feeling pain. We don't know about suffering because that's more on the psychological, but at least pain. And it's, if there's a nervous system, that animal is going to feel some sensation, right? So typically in the lab, the very first ones who get it are the bacteria. And then, well, usually that's the step, you know, if we're looking toward clinical trials on human. Bacteria, the fruit fly, the mouse, and then the human, okay? And for the human, we need consent, unlike the other three. But anyway, we could test some things on non-humans before we test it on human, and that is ethically acceptable. Of course, not that we're going to be killing them uselessly, or that the experiment has to be designed well and correct, the best possible, so as to minimize the impact on the animal that is being used, all right? But at the end of the day, the mice are grown in the lab, and you know, I think a female drops a litter every 3 weeks or so. And the litter could have 4, 5, 6, 7. So they're quite abundant that way. But still, there has to be a justification. And believe me, the protocol for sacrificing or euthanizing a mouse in a lab is pretty elaborate and has to be followed. And if that protocol is not followed, not only from the scientific side, but also from the ethical side, that lab can be shut down, okay? Because these are strict protocols. Again, when I was doing that research the year before going to Purdue, typically the labs have, the basement will be for raising or maintaining the animals, the lab animals. Mice and rabbits are the ones that are used mostly in the medical setting. And I remember down there, if the female can drop the litter any moment, and immediately after she gives birth, the male has to be taken out because dad will come around and start eating up the babies. Yeah, they're pretty voracious that way. They have no paternal sense. And so someone has to be on duty watching that female. And as soon as she drops the litter, then someone has to go in and literally physically take out the mouse. If it's Sunday at 3 a.m., it's got to be done, because otherwise by the next morning, the litter will be gone in the tummy of the father. There's no mercy. Anyway, so this is the first level of distinction from the bioethics ethical perspective, where the stem cell research is on human, on non-human, okay? Coming from the human or the non-human. So then the second level, of course, is within the human, if it's embryonic or adult stem cell. In fact, this adult I should put in quotations, because Adult is any born for the purpose of stem cell research. In other words, even babies have stem cells in them, right? There's an increasing sources of stem cells being discovered in the body. First, it was things like the pulp of baby teeth. You know how we shed our teeth, right? The pulp of baby teeth has stem cells. Hair follicles have stem cells. Adipose tissues or liposuction would have stem cells in it. And bone marrow, for example, has stem cells. You know, they're precursor, they're hematopoietic cells that make up blood corpuscles. But even now in the blood itself, scientists have discovered stem cells. They're not a lot, there are a few, but they're there, which means that even just from a transfusion, one could obtain stem cells from one's own body, all right? They're called autologous. We'll get to that one. But I put adult in quotations because basically means from the born forward, all right? Of course, if the born person donating the stem cells is underage, then someone has to consent. Typically, the parents will have to consent, but the idea is that it's not harmful or minimally harmful for the benefit that the person is getting. So the harm may be as little as just a needle stick, right, to obtain them. But there's an increasing sources of stem cells that are being discovered in the body, in the human body, and that's a good thing. And you'll see why in a minute. But going back to the embryonic now, the challenge with the embryonic stem cells from the bioethical perspective is that they come from where? They come from the ICM, all right? If you remember, the ICM is the inner cell mass. It's what becomes the embryo proper. This is a micrograph, a photograph of a microscopic photograph of a blastocyst, a human blastocyst. And as you can see here, we have two different types of cell layers already. We have an outer layer of cells that forms it's known as a trophoblast, right? Which is like a ball, like a hollow ball. This is the blastocyst. Remember, in diameter, it's about the same size as the original ovum, 10 to 15 microns in diameter. But then there's this little mass of cells that are more compacted, and they're just unimaginatively called inner cell mass, or ICM. Sometimes you also see it referenced as the embryoblast, because the embryo proper comes from here. Remember, the embryo proper forms from here, and the trophoblast becomes what? Think functionally, the trophoblast, which is in more direct contact with the uterus, a normal implantation. The ICM or the trophoblast? Trophoblast. The trophoblast is the direct contact with the endometrium of the uterus, right? And therefore, the trophoblast will become the placenta on the embryonic side. There's also placenta on the mother's side. You know, both are vascularizing, are making connections between the capillaries. So that placenta, that very complex filtering organ, is formed from the trophoblast. Whereas the ICM is going to elongate and form the gastrula and then undergo gastrulation, the 3 germ layers, endoderm, mesoderm, and ectoderm, and then continue into organogenesis, forming the various organs of the body and the different regions, right? From the 3 germ layers are the ones that produce all of the organs in the body. So you notice that because we have 2 distinct cell layers there, 2 distinct tissues, differentiation has already occurred. And therefore, it has gone from totipotent to pluripotent, okay? The potency of the cells. Anyway, it is the ICM that is plucked out and put on a petri dish to become the human embryonic stem cells. Okay, bless you. And that's the big ethical issue. Of course, to pull out this ICM, the trophoblast has to be disaggregated. The blastocyst has to be teased apart with sterile needles and so forth. And so once we pluck out the ICM, in effect, we are killing that embryo, that early blastocyst, right? And so to produce the human embryonic stem cells, a human embryo has to be killed. They talk about destroying the embryo. That's when they want to admit it. But even destroying is a euphemism because we destroy things. We can destroy a chair, a car, or an airplane, but humans are not destroyed. Humans are killed. And so truly what is happening here, the moral object is that the human embryo has to be killed in order to obtain the stem cells, to obtain embryonic stem cells. And yes, they have tremendous potential, and that's why they are the gold standard, right? They're what is known as the gold standard, but they're gold standard theoretically. You'll see why. Let me back out for a moment and do a little review of cell potency here, just for background. There are 4 levels of differentiation, if you will, beginning from non-differentiated cells, undifferentiated, to fully differentiated cells, okay? 4 stages. So the first one, they're called totipotent. Again, the little added value of the course is a little Latin or Greek. So toti is a reference for all, todo, total in español. So totipotent, they have full potential. All of the cells that are totipotent have full potential for what? Full potential to become the whole person, the whole human being. And the first 2 stages are totipotent. For example, what about the zygote? Zygote has to be totipotent because we have a single cell, right? And from that one cell, the entire embryo will develop. Not only human, any embryo, even plants go through a zygote, right? So the zygotic stage is totipotent by definition because the entire individual of whatever species will come out of that one cell, totipotent. It turns out that the morula is also totipotent. In other words, they talk about Spemann, Hans Spemann, remember the German from last century who was taking frog embryos and teasing them apart. Frog morally, he would take a morula from a frog and tease the blastomeres. Each cell, individual cell is called a blastomere, right? And he would disaggregate the morula and just let it sit in the water there. And soon enough, each individual blastomere would start undergoing mitosis. And each one of those would become a little morula, and then he would let it go some more, and each morula would become a blastula, and eventually a tadpole, and eventually an adult frog who could mate and reproduce, and so fertile offspring, right? So basically, that simple little experiment proved that even the blastomeres of the morula are totipotent, which is another qualm that one would have bioethically. Remember when we talked about in vitro fertilization, how they do pre-implantation genetic diagnosis, PIGD, right? Pre-implantation, before implanting, they would pluck out a blastomere from the morula, send it to the lab for analysis, for chromosomal analysis, and see at least a karyotype to see if it was full number of chromosomes and whatnot. Plucking out that one blastomere may possibly be generating a new human now, because that new blastomere may revert to becoming a zygote because of its totipotency. So that's another one. I mean, and there's the possibility, okay? And so if there is a possibility of generating a human being like that, which in effect would be a clone, and then sending that for lab analysis, which is going to destroy that cell, we may actually be killing that human being. And so that's another subtle but real reason why PIGD is again unethical. Anyway, these are totipotent, the zygote and the morula. Then the third stage, remember, is the blastocyst. I was just talking about that. And because we have cellular differentiation here, this is the first level of cellular differentiation, right? What are the two layers? The trophoblast and the ICM. And because we have cellular differentiation, now the cells that make up a blastocyst are no longer totipotent. Now they are pluripotent. Pluri meaning many, but not all. In other words, that any one of these cells may become many other cells in the body, but not all of them. For example, the trophoblast is not going to become the embryo proper. And the ICM is not going to develop a placenta by itself, okay? And so some organs and tissues are already being discarded by the first level of differentiation, of cellular differentiation. And when you think about it from the cellular perspective and from the perspective of tissues and organs, functional tissues and organs, that's really what the embryonic development is all about. Those 9 months is about cellular differentiation to get, a morula or a blastocyst or a gastrula eventually to develop a functional liver, a functional lung, a functional heart, a functional brain, right? Those are differentiated organs, so that the brain does not function like a liver, and the liver does not function like an arm, and the arm does not function like a spleen, okay? Those are differentiated. So pluripotent means that it can become many cells of the body, but not all right? And then the next stage, more further differentiation after the gastrula. Here's gastrulation with the 3 layers, the endoderm, the mesoderm, and the ectoderm, all right? This is a yolk sac underneath, which we have a little tiny yolk sac that is resorbed and used up early on before the placenta is established. Anyway, after gastrulation, eventually we move into the fetal stage. After 8 weeks, remember we have the little embryo, all of the major organs are in place, but they're tiny because the whole embryo is about 1 inch long. 8 weeks, 2 months. And then for the next 7 months, that little embryo is mostly growing in size, okay? But the basic development, the basic differentiation, cellular differentiation already happened in the first 2 months, first 8 weeks of our lives. Just amazing, very, very fast. Why? Well, because those organs need to function. They need to get started, and the liver needs to start doing its thing in digestion. The fetus is digesting, right? The fetus has circulation, so the heart has to be pumping and so forth. The last organ to develop, actual system, but mostly the organ is going to be the brain, because it's the one that is needed later, all right? Especially the cerebrum itself for cognitive thought. The lower brain develops earlier because of the autonomic system needs to get functioning, right, in coordinating all of the other organs of the body. So we have this gradual process of differentiation until we get to fully differentiated cells. I just put one neuron here simply as emblematic because the neurons are probably one of the most specialized cells that we have in the body, okay? And they essentially do no other function than neuronal function, transmitting neurotransmitters of signal. Okay. So we have the 4 stages basically of cell potency. So going back here to the ICM is pluripotent, all right? And that's why it's considered the gold standard, because in nature, from the ICM develops the entire embryo. And therefore, at least theoretically, any organ or tissue may be able to be regenerated with these embryonic stem cells, all right? That's why, again, they're called the gold standard, because from these embryonic stem cells, in principle, any organ or tissue of the body could be regenerated. We have about 220 cell lines. A cell line is a cell, like the precursors of the tissues of our body, all right? the organs and tissues, and they're called cell lines. And we have about 220 cell lines in the body that make up all of the organs and tissues of our body. So the idea was to develop about maybe 100 to 150 cell lines, and with those be able to regenerate essentially any organ or tissue of the body that was either injured or became diseased, pathological, all right? Including, for example, cancers. So the idea was with cancer, like we talked about last week, a tissue or an organ is removed, the mass is removed, and then that organ, the remainder of that organ could be regenerated with stem cells. That was the idea. That's the theory in what is known as regenerative medicine, okay? Regenerative medicine. So it's a field that's very promising, still experimental, but the ethical challenge there, of course, is that to obtain embryonic stem cells, the embryo has to be killed. All right. And therefore, from a principle of bioethics, we take exception to that. Now, the good news is that there is an alternative source of stem cells that are actually working better than the embryonic stem cells. And these are known as adult stem cells, like I say, from the born forward, simply because they're no longer embryonic, okay? So we're not going after the ICM, rather different organs and tissues of the body that lodge stem cells. So they are multipotent instead of pluripotent. You would think that this is a disadvantage, but it's actually an advantage. You'll see why in a minute. But they're not pluripotent like the embryonic stem cells. They typically are multipotent. In other words, they are within a particular organ system. So they will be either from the circulatory system or the digestive system or the vascular system, all right? They're within an organ system, but they generally don't jump from one organ system to another. And that's why they're called multipotent as opposed to pluripotent. They're a little more specialized than the pluripotent. Okay. An advantage is that we can do autologous transplants. So autologous is a reference to self. Auto is a reference to cell, like the automobile is the one that drives itself, has a self-propulsion, right? The motor. So autologous transplant means that we can take tissue, we can take stem cells from one part of the body and implant them into another part of the body. For example, take stem cells from the blood of a patient and inject them into the knees to try to alleviate arthritis, things like that, all right? And so there is no immune rejection because they come from the same patient, all right? That's a definite clinical advantage. There's no immune rejection. As I go here, I need to mention two big disadvantages of the embryonic stem cells, aside from the fact of the killing of the human embryo. These two disadvantages are disadvantages in practice, biological, medical disadvantages. Think about that. First disadvantage, the ICM necessarily is a different individual from the patient who's receiving the stem cells, right? And so there is going to be an immune rejection from the patient to those stem cells because they're not the self. It's like an organ transplant, even though it's from human to human, because of the genetic fingerprinting that we have, there is an immune rejection. An organ from another person, from another human being, is seen as a foreign object for my body. And my body will mount an immune rejection to that organ, even if the organ comes from another human being, all right? And that's the whole issue of matching and compatibility to make the match so that the immune system will not reject that, that organ. Well, the same argument here, because the ICM is necessarily coming from another individual human being, and therefore those cells are seen by the patient's body as foreign, and therefore the patient's body will now mount an immune rejection against those embryonic stem cells. So that's a big negative. What does that mean? That can be overcome because just like with organ transplants, the patient could be put on immunosuppressors. Immunosuppressors tone down the immune system. They don't shut it down altogether because then they would be susceptible just to breathing air and becoming infected, right? Because the air is not sterile. But they tone down the immune system so that it would not kill the embryonic stem cells, all right? But it's not the ideal because the patient has some kind of illness, and now on top of that, they're also immune suppressed, so they have to be very careful about getting an infection. What would be a common cold for us could become pneumonia for them, right? So that's a big negative. But another one that is even worse is that precisely because the stem cells, the embryonic stem cells come from the ICM and they are pluripotent, they have too much potential actually. So when these experiments were done in mice, before going to humans, the typical result is that those embryonic stem cells start growing undifferentiated and they eventually turn into tumors, cancerous tumors. So now a patient with a pathology getting embryonic stem cells will, on top of whatever pathology they have, they're going to develop also tumors from the embryonic stem cells because they have too much potential, all right? It's a simple little analogy would be if I have one of those transistor radios, I don't know if you remember, I remember transistor radios, they have little batteries, okay? But the battery, instead of being a 1.5 volt battery, let's say it's a 150 volt battery. Same size, but 150 volts. That's the potency, all right? So if I put that battery inside the radio, it's gonna fry the radio, it's gonna frazzle it. Too much potential, right? And so that's what happens with these embryonic stem cells. They actually have too much potential, all right, by being pluripotent. And so what I mentioned as the theoretical gold standard in practice is bad news because now they're generating tumors on the patient instead of healing the patient, okay? So the fact that the adult stem cells are toned down into multipotent is actually a good thing. It's positive clinically, all right? And so to this day, there have been, and this research has been going on for about 2 decades now at least, there have been no actual cures from embryonic stem cells in the human. And there has been an increasing number of cures with adult stem cells. And so just on the practice of it, the adult stem cells are coming out better than the embryonic. There's a website here that early on started tracking the success and actually started collecting and storing the scientific articles that were reporting success, either on embryonic or adult stem cells. And it's a group called Do No Harm. It's a number of doctors and philosophers and theologians. And they have a website called just Stem Cell Research, single word. I want the website. I think it's .org. .org? Okay. Because they have been documenting this for quite a few years. There it is. Okay. And they're collecting all of the articles that have cures at different stages from the adult and from the embryonic. And to this day, the conclusion is there's 0 from the embryonic and 73 from the adult. Here it is. Show you just a list. So here is a list of the adult stem cells in various stages of clinical trial, number of cancers, autoimmune diseases, cardiovascular, ocular. regeneration of the macula, for example, or cornea, immunodeficiencies, neurodegenerative diseases, anemias, wounds and injuries, particularly in wounds and injuries. This is some of the most successful areas. Okay. So it's a nifty website. It's this group called Do No Harm. And they do that. They scanned scientific literature for successes in both. There has been none in embryonic so far. Geron, I think Geron Corporation tried getting a clinical trial going a few years ago. And finally, they had approval from the FDA for their protocol. And then what happened was when they started on the clinical trials, some of the preliminary studies began to look a little funny and discouraging, and abnormalities growing. And so they shut it down because they were going to get sued basically for inducing cancer in their patients. Geron is one of the oldest biotech corporations, maybe goes back to the 70s or something like that, back in, I think it's in California. So there are basically no trials with the embryonic, and after all that hype and money, millions of dollars that have been poured into embryonic stem cells, zero results. But it's encouraging to see that the adult stem cells are producing results with a number of distinct advantages just on the biological medical side of things, aside from the ethical considerations. And so what is needed for adult stem cells to be ethical? It's so obvious that people just don't think about it, but we covered it earlier on, which is from, say, if it's going to be autologous, from the patient, consent to take out his or her stem cells to be used for himself or herself. And if it's going to be donated, right, then again, consent from the person who's donating the stem cells. That's all that's needed is consent. And so minors, someone has to consent on their behalf, but there has to be a serious reason for involving minors in any kind of research. Okay. All right, another source is the IPS. Have any of you heard of the induced pluripotent stem cells? Induced pluripotent, right? Shinri Yamanaka. I remember Yamanaka, he's a Japanese researcher. And it was when I was doing my genetics at Purdue in the beginning of 2000s. Yamanaka was doing embryonic stem cell, human embryonic stem cell research originally. And as he was looking at the human blastocyst under the microscope to disaggregate them and to tease out the ICM, you know, he thought to himself, because later he was interviewed, and he thought to himself, these blastocysts that I'm looking at under the microscope, I'm paraphrasing, look very much like my daughters used to look like when they were that stage. Those were his thoughts. In other words, he was an honest man, because he has daughters, you know, and he recognized that those blastocysts are human blastocysts and that his daughters were that same size and form when they were at that stage of development. And so he kept saying to himself, there has to be an alternative to this. There has to be an alternative to destroying these human embryos. Okay, and that's in the early 2000s when the controversy was raging, because again, the embryonic stem cells were thought to be the gold standard. And so he says, what if we take differentiated cells and we try to undifferentiate them? We try to de-differentiate them, go backwards, okay, which is totally against what nature does, but it may be done. And so he started working on this with a team of researchers in his lab. And they were working very hard, long hours, and other labs in the world started getting onto it also. They were looking essentially for factors, for what is known as regulatory factors, regulatory proteins in early development, okay? And it came down to about 3 or 4 regulatory proteins. They are the ones that are in charge of cellular differentiation early on. And by either taking those out or varying their concentration, he got differentiated cells to undifferentiate, to revert back to pluripotency, but not totipotency. So he did not end up making embryos, right? He stopped at pluripotency, which is fine because we don't have a full human embryo. We just have pluripotent cells, okay? And so he did it with Blastocyst, human fibroblasts, which is part of the muscle tissue, all right? And he was able to undifferentiate cells to, from human fibroblasts to pluripotent, and then redifferentiate them into other organ systems. And that's why they're called induced. The I stands for induced because this doesn't happen in nature. This happens by tinkering with these cells with the regulatory factors, the regulatory proteins that are in charge in early cellular differentiation. Okay. For that, he got the Nobel Prize. He worked very, very hard at it, and he did it precisely because he was inspired by the ethics of it. Even though he was a very hardcore scientist, he had scruples about killing these human embryos. And so he said there has to be another way, and he found it. The challenge with the iPS is that because it's an artificial process, right, it's still not sure if these undifferentiated cells that are then redifferentiated, if they maintain their characteristics, their full characteristics of that differentiated tissue or not. So still some research going on there. Plus the vectors that are used for injecting these factors, these developmental factors into the cells are essentially viral capsules. And there's a concern that because this is done gunshot, right? You cannot take each individual viral capsule and clean it out of its DNA or RNA and then fill it with the regulatory proteins, every single one, because you're dealing with billions of viruses. So it's a gunshot thing, and therefore the fear was that some of those viruses may still have their own DNA or RNA intact. I will be injecting that into the cells, even if it's a very small amount, thus contaminating the entire lot of the cells that are being differentiated or undifferentiated. So there was an issue with that on the delivery, because the viruses are essentially used as super macroscopic needles or syringes to inject, literally inject the factors into individual cells or tissue. Okay, so that's a work in progress, but at least it's from our century for this, from the 2000s forward, all right, the last 20 years on stem cell research. And then there is the other one, the famous cord blood, which comes from the umbilical cord, all right? Now, umbilical cord is connecting what to what? The umbilical cord, the placenta to the fetus, right? When the baby is born, what happens to that cord? Cut, severed, tied on the mom's side or the baby side, the knot is pushed in and that becomes the navel. And on the placenta side, it's put into a biohazard bag and incinerated, so it becomes biomedical waste. But the placenta is very rich placenta itself has stem cells in it, okay? But the cord blood inside, there's a little bit of blood inside the umbilical cord, and that cord blood has also stem cells. And there are stem cells that are from the baby, okay? And so those are also a very good source of stem cells, which technically are adult, you see, because the baby is born already, even though they are very high quality, they're pristine because they have not been exposed to UV or anything like that. They are just coming out of the womb, so they are really very plastic, dynamic, and they're called mesenchymal stem cells. Mesenchymal stem cells, and can produce any of these tissues, typically in the mesoderm and endoderm, liver, fat, muscle cells, cardiac muscle, nerve cells, cartilage, reconstruction of cartilage and bone, all right? So mesenchymal stem cells have a lot of potential. And are they ethical or not ethical? Yeah, does it harm the baby in any way? No, this after the baby is born and the cord is severed from the baby, so it doesn't harm the baby in any way. It's true that the placenta is a vital organ, but only until birth, and after birth is discarded, okay? So those stem cells, and that umbilical cord and that cord blood would otherwise just be thrown out. So certainly they're ethical, they're good to use. And still, there is a lot of ignorance around this. Going back to that same hospital, for some reason, I'm hovering around the Boston experience on this lecture. But I remember while I was at that research hospital in Boston, the year before going to graduate school, it's St. Elizabeth Medical Center. So it's the Catholic hospital in Boston. It's a huge hospital. And I was saying to Louis, that's it was the teaching hospital for Tufts University Medical School. And I asked one of the nurses there, while I was doing some research, one of the nurses in the ICU, in the NICU, neonatal ICU unit, I asked her, do you save the cord blood here for stem cells? And she said to me, oh no, we don't do that here because this is a Catholic hospital. In other words, like say there was some ethical issue about this, right? And this was a nurse, this is a NICU nurse, okay, neonatal nurse. And so it's amazing, it really struck me the level of ignorance, with all due respect, that she really didn't understand the process, that it doesn't really harm the baby at all. All you need is consent from the parents, but there's no ethical issue. She was just thinking of stem cells, you know, stay away from stem cells, because in her mind it was embryonic stem cells, you see? But there's unfortunately a lot of ignorance about this, even in the medical profession. But an increasing number of couples now, when they have their children, they're saving the cord blood. And that's a good investment, because those cord blood, those stem cells are pristine and have an immune compatibility with the baby, with the child, as the child grows. Now, of course, the ideal is that those stem cells will never be used, right? So in other words, that the child will never have any kind of injury or disease or anything, so you don't need them. But in case you need them, they're there. That would be how they preserved. Cryopreservation, they're cryopreserved, okay? And that's it. So it's an increasing number of couples are getting into this. And I think personally, it's a good thing. It's a good healthy alternative. The downside is that typically medical insurance doesn't cover it. So they have to pay for the initial expense of the extraction. You can actually buy a kit online. You buy the kit online, which consists mostly of a syringe to extract, and then the vials in which to keep. It's actually sent, the blood is sent to the lab for separation, and then the stem cells are collected, and that's what's cryopreserved. But there's an initial expense of the kit there to extract them. And then the maintenance on a monthly basis or yearly basis, whatever is being charged. You know. So there's some expense. And as far as I know, insurance is not covering it. So it has to be out of pocket from the couple. But that's a very good, healthy alternative to the embryonic stem cells is cord blood stem cells, all right? Or just known as, cord blood stem cells. Okay, that's as far as the different types of stem cell and research. Now we're gonna shift, well, any questions or comments on any of that, the stem cell research? Little by little, this is becoming kind of historical because since the embryonic over several decades now are not showing any progress due to those two big issues of the immune incompatibility and also the fact that they generate tumors typically, then they're being put aside. And yet some scientists don't want to give up and keep researching on them. But now the embryonic stem cells, precisely because they cause tumors, they're being used as models for cancer research. Okay, so kind of in a roundabout, the embryo keeps getting attacked because now these embryonic stem cells, since they cause cancer, well, then let's use them as models for cancer research. It's unfortunate, but you know, not everyone has the principal bioethics system that I'm talking about. So they justified saying, well, if we find a cure for cancer, then the means are justified. Okay, let's move on to cloning if there are no questions or issues. It can be seen as reproductive cloning or therapeutic cloning for the purpose of functionality. What is it being used for, okay? So functionally, what is the human cloning being used for? To generate another human being or to produce embryonic stem cells, precisely. So again, this one is a little dated because of that, but let's look at it. These are the two alternatives regarding what the cloning is being done for, what the purpose. All right, there's a fancier word for cloning, which is somatic cell nuclear transfer. You know that in science, we don't like people to understand what we say or do, so we make up these fancy phrases, right? But it describes the same thing, it's describing the same thing. Somatic cell nuclear transfer. All right, so what do we know about a somatic cell? Somatic cell is not a gamete, it's diploid instead of haploid, right? And therefore, the nucleus of a somatic cell has the full complement of chromosomes, in our case, 23 pairs, okay? And somatic is a reference to soma, and soma is a reference to body, the body. I know, do you ever see the movie Soma? No, oh, it's a bad movie. It's about bodies, human bodies that were being floated in the room, and anyway, it's a bad sci-fi. There's a video game called Soma. Okay, maybe it takes after the movie.
It's about mental transfer between bodies.
Oh, there you go. Okay, yeah. So it's a reference to body one way or another. Okay, so somatic cells are all the cells in the body except the gametes, right? Because these are all diploid cells, meaning that the nucleus has a full complement of chromosomes, has the 23 pairs. All right, so, and what's the first somatic cell in the body? The zygote. After fusion, after syngamy, diploidy is restored. And so the first somatic cell is the zygote, and then from there on forward. So somatic cell nuclear transfer is exactly what it says, taking the nucleus of a somatic cell and transferring that nucleus into an enucleated egg, into an enucleated ovum. So you just have the cytoplasm of the ovum. When you have the cytoplasm of an ovum and you put in a somatic nucleus, what have we manufactured? We have manufactured a zygote, right? Functionally is equivalent. So SCNT is the acronym for cloning on whatever. And the first mammal clone was Dolly the sheep, exactly. We're going to go there. Okay, so it just describes here. It's basically a two-step thing. You need an ovum, enucleate the ovum, and then renucleate that ovum with a somatic nucleus, all right? And it's essentially what you're doing is you're transforming a gamete into a somatic cell, into a diploid cell. But because of all the factors that are in the cytoplasm, meaning the regulatory proteins, those proteins will start working on that nucleus, on the DNA in the nucleus, and will start expressing the nucleus, the nuclear DNA, progressively as if it were a zygote. And that new zygote will now undergo development and become a morula, a blastocyst, a gastrula, et cetera, all right? So that's essentially what happens there, because the regulatory factors are precisely in the cytoplasm of the ovum, just waiting to act on the new diploid nucleus. All right, there's a reference there. You can take a look. Basically, let's go to Dolly for a moment. There's Ian Wilmot. Wilmot is the researcher who was at the Roslin Institute in Scotland, who's the first one to, was able to engineer, to manufacture a clone, and the first mammal in the lab at the Roslin Institute, okay? 1996. So this is a process. We need an ewe, right? An ewe is a female sheep. And we need here, this one is the ewe because here's the egg. Enucleate, take this nucleus out. Then we need another, it could be another or the same animal, but we need a somatic cell basically. And we need the nucleus, the donor nucleus is somatic, is diploid, remember, right? Full complement of chromosomes. And they are fused together. There are various techniques. One technique is to inject the nucleus into the ovum. Another technique is by an electric shock to actually, they don't even bother taking out the nucleus now, they just take the whole cell and fuse it with a little electric shock into the enucleated ovum. It's essential that the ovum be, that the haploid nucleus be taken out of the ovum, all right? Otherwise you're gonna end up with a triploid. You don't want a triploid. Okay, so once the fusion occurs, now we have the cytoplasmic egg with the diploid nucleus. So in effect, we have now a zygote, which will start differentiation, morula, and then the blastocyst stage will be implanted, just like IVF, right? Will be implanted into the sheep and wait a few months, I don't know how long is gestation for the sheep, until the baby sheep comes out, okay? So you can see that you can use up to 3 different sheep, but you can also narrow it down to 2 or 1. In other words, the same sheep, the same female sheep can be the one who donates the egg, donates the somatic cell, and then gestates, incubates the embryo, okay? They did it from 3 different sheep just to prove the principle that it can be done by different sheep. But the proof of principle, they had to wait until the clone lamb grew up and did what? To see if it was functional and working. Exactly, reproductive success. All right, so they had to wait until Dolly grew into adulthood And then they mated her and she produced fertile offspring. So that was the final proof of pudding, a little bit like Louise Brown with in vitro fertilization, okay? And that was Dolly.
Didn't Dolly die premature?
Yes, exactly, good point. And that's what happened. So it turns out that Dolly was actually euthanized. Dolly was killed because Dolly started developing, even though she was still young, she was like a young adult, she started developing diseases of the old age, arthritis, the joints, and some other issues that typically are associated with older mammals, okay? And they realized that she was actually in pain. I guess she was having difficulty even just getting around. So they ended up euthanizing her for humane reasons. And that's one of the issues that was used for scientists to speak against reproductive cloning. In other words, to use human cloning for reproductive purposes, for generating a new baby, all right? Because, so after Dolly, many other mammals were cloned. A cat, I remember the cat was called Copycat. It was the first clone cat, Copycat. And then the other mammals, pigs, then it got into the animal husbandry, of course, pigs, cattle, etc., to enhance, because you can enhance, you can do artificial selection, right, at the genetic level. If this sheep is producing a lot of milk or a lot of, let's say, good wool, you want to maintain those genes, and you can clone that sheep, you know, from a single cell into other sheep that make the same wool, the same good wool or abundant wool, whatever it is. And so in animal husbandry had a lot of market value, the cloning thing. So quickly, mammalian cloning took off, all right? And the question was, should we clone humans? For reproductive purposes, no, because invariably what was seen is that these clone mammals would age prematurely. It was like a standard thing. They would age prematurely, and they thought that would be cruel then, because for when we go to the human, cloning that human, we would know beforehand that on average, this human is going to age prematurely. And then what are we going to do? We're going to euthanize the human also, right? Yes.
What about the offspring of Dolly? Did they exhibit the same issues?
I don't know, to be honest with you, but I really don't know. I don't know. No. Yeah, it's a good question. It would be interesting to see. Maybe we can all look it up and see if Dolly's offspring also aged prematurely. Because then it means that it's being passed on, right? Inherited. So that was one of the reasons for scientists to speak against reproductive cloning, because of this premature aging. Another thing, by the way, which is not really mentioned much, but it took about 250 trials for Wilmot to manage to get a Dolly, all right, for an embryo to implant and just stayed long enough to be born alive. So it's about 250 trials. So a lot of trials. And that seems to be kind of a standard also for mammals in general. Not all, the vast majority don't work. They die at some point. Either the fusion doesn't happen correctly, or the development into the morula, blastocyst, doesn't happen, or even at implantation, the blastocyst, most of the blastocysts don't implant, all right? It just takes a lot of trials, an average of about 250. So again, when you translate that to the human, biologically speaking, we're mammals. So we could expect the same average, more or less, 250 trials to get one clone human, all right? That means 250 deaths to get one clone human. So there are a lot of issues there for which the scientists spoke against reproductive cloning. However, for therapeutic cloning, they were all in favor, okay? Because the idea in therapeutic cloning is to generate the blastocyst right, from the somatic cell nuclear transfer. And once we have the blastocyst, now extract the ICM for embryonic stem cells. So it was seen as a cheaper way of obtaining embryonic stem cells by doing this cloning. And according to them, you know, they're not killing human embryos because these clones are being manufactured in the lab anyway from the nuclear transfer. However, the fact that once we have a zygote who's developing to a morula and a blastocyst and has a human genome in it, we have to assume that that's a human blastocyst, okay? So the idea about therapeutic cloning was to, as a source of embryonic stem cells. Either way, on the cloning, I think it should be pretty obvious that from the principal perspective, we would be against both types of cloning because it's just an excessive manipulation of human life. And it's a life that cannot defend itself. And therefore, we just don't have a right to manufacture them in the lab like this. One last thing. So, okay, so there's a diagram for therapeutic cloning. As you can see, it parallels the one on stem cell research. The idea is to generate human blastocysts and then obtain the embryonic stem cells from the inner cell mass. Okay. Reproductive cloning. There are two possibilities. One possibility would be one woman is an egg donor. The enucleation happens. Now we have an enucleated ovum, which is fused with a cell from who is going to become the mother. So the egg donor doesn't have to be the mother necessarily, just like surrogate motherhood, right? It could be a third party, or it could be the mother who is going to gestate that baby also. So just like with IVF, in reproductive cloning, that child can end up with up to 5 parents, right? The woman who donated the egg, the person who donated the somatic cell, could be another woman, could be another man, just a somatic cell. And then the woman who gestated that embryo, and finally the couple who paid for the whole procedure. So it's kind of parallels IVF. It's just that there's a further manipulation here by the cloning procedure by the nuclear transfer itself. Okay.
People really don't want to adopt, do they?
Right. Well, you know, the reality is that it's very hard to find adopted babies to adopt in the US, like healthy babies, all right? You have, fortunately, you have crack babies and babies like this, but it's hard to healthy babies. Plus, this whole thing of the genetics, at least it'll be part of my genetics, right? Because I could be the egg donor or I could be the cell donor. Like the father could be the cell donor, the somatic cell, and it's my genetics. So it's just a mentality. It's sad, but it's really a manipulative mentality, whether they realize it or not.
That's a lot of work.
It is a lot of work. But, well, this industry is a boutique industry, right? And they cater to boutique industry, meaning that they cater to the rich, to whoever wants to pay. And who gets into this? The entertainment industry and athletes and people who have contracts of millions of dollars that can afford all this because insurance is not going to pay for it. But they want to do these extravagant things. It's sad, but it's a lot of manipulation. One last little tidbit. If there are no questions or comments, trivia question. Why is Dolly called Dolly?
Oh, she was named after someone. I just never remember.
Dolly Parton, the famous singer and actor, Dolly Parton. You know Dolly Parton? Don't tell me, you guys are so young, you don't know Dolly Parton? She's one of the greatest, I have a lot of respect for Dolly Parton as an actress. Yeah, she's elderly now. She's gone through a lot of facelifts and so forth, but that's Dolly Parton, right? Remember her? She's done a lot. Oh, Steel Magnolias. You want to see a good movie now? Of three women. Steel Magnolias. Yeah, that's Dolly Parton. Way back when she was young. Now when she's got all the facelifts. Very good singer. Yeah, she's got a lot of the golden records there and so forth.
Sung with all the famous country singers like
Right, country music. Country music. What is that in Tennessee? What is the Grand Ole Opry? Grand Ole Opry, exactly. Great actress, great lady, great singer. But it was tongue in cheek. So why? What's the reference, right? It turns out, and this is from Wilmot's own words, because I remember when this was going on. Wilmot, I guess he was a fan of Dolly Parton. And so where did the somatic cell come from? The somatic cell of the donor, right, the diploid cell, came from the udder of the sheep. And what is the udder? U-D-D-E-R, the udder. The udder is the mammary glands of the sheep, of cattle, all right? And so it was in honor of Dolly Parton because of her prodigious mammary glands. This is for real. Tongue in cheek, okay? But that was Ian Wilmot.
That's extremely specific.
It is very specific. It's actually offensive. I find it offensive, but it was, yeah, the prodigious mammary glands of Dolly Parton, because the donor cell came from the udder of the sheep.
Whoever did that had an unhealthy obsession.
Yes, very. But that was, yeah. That was it.
I couldn't find anything about Dolly's
Offspring?
but I did find that 4 sheep produced from the same cells as Dolly were raised differently from Dolly and are currently 9 years old.
Oh, really?
Yeah.
Oh. So the nurture could have had a lot to do with it.
The hypothesis seems to be that they should be raised like in outdoor conditions, free to roam, space, and things like that. And that Dolly was more controlled, and that may have contributed to like the movement.
I see. Okay. That could have accelerated the process. Interesting.
And also, I think the process of cloning, I think, still has like an 80% efficiency rate.
Yeah.
Which is why they don't really. Like the thing I was reading said, yeah, it's not equipped for like human use because.
Right. There's a lot of trial and error.
Roughly 20% of your people might die prematurely.
Right. Yeah.
But yeah, it has progressed. I mean,
Right. Yeah. Some of this is a little historical. I think that scientists have walked away a little bit from cloning, at least for the human, right? And also because the embryonic, so even for the therapeutic cloning, which was for the purpose of generating embryonic stem cells, if those human embryonic stem cells are not giving results anyway, then why go there? Why invest the money into that? Because it is expensive research if they're not getting results after decades. And in the meantime, the adult stem cells, which are much cheaper to obtain or ethical, etc., they're giving results. So let's put the money into that, right? So here's one case where principle bioethics, let's say, has won the day, but not because of the principle, simply because of the fact of the practice that the other ones are not working. Okay, anyway, I think that you may run into stem cell research or cloning at some point in your future studies, so I figured I'd insert this lecture. Okay, that's about it then. Questions or comments from Lisa, are you the only one out there? Yeah, I think so. Okay.
Oh, I'm okay.
Okay, thank you. Then we'll meet up Friday. Mhm. Okay, let me stop this.